One of the most rewarding parts of being a graduate advisor and being a mentor is when students really develop and you see them starting to reach their potential. Josh always had great ideas, and over the last 4 years as a graduate student, what's happened is now he's turned into this guy who takes these great ideas that he has, and he can go through all of the steps of designing experiments, developing a project, and seeing this all the way to fruition. And so what we're going to see today is kind of the culmination of this in his PhD defense, and he's going to talk about a range of different projects looking at bioinformatics. He's done metabolomics, he's done proteomics, he's done microfabrication and machine learning. So enjoy. So all right, well, I think all of the pieces are now in place, and thank you all for showing up this morning and braving this nice Montana weather. I have to admit, if it wasn't for this, I would have been really tempted to be skiing today. So the fact that you're all here, that really says something.
But I Skype in from the ski lift.
Yeah. But I think we're all in for a treat, as Josh kind of gives us, I think, kind of a look at all that he's accomplished throughout his PhD here. Josh has been, a wonderful student to have in the group, and his research, as you'll see, has covered not only different topics, but really he has 4 first author papers, or 3 that are published and 1 that's being submitted to JAX, that are in completely different research fields, not only topics. This is really, I think, quite amazing, and he's really been the driver for each of these projects. And so I think we'll get to kind of see some of that breadth in his work today. And with that, I'll just let you go at it, Josh.
Thanks, Brian. So yes, thanks everyone for coming and braving the weather. And I'd just like to start by saying, because of the breadth of my research, it was really hard to come up with a title to fit and encompass all of the research topics I'm going to talk about. So I eventually landed on this, the fossil viruses, redox paradigms, and predictive metabolism from a systems biology perspective. So systems biology, for those of you that don't specialize in what we do, is what is it? And it's a loosely defined term. It's an interdisciplinary field, and it tries to generate a holistic perspective of life or biology. And interestingly enough, where it finds its roots is in general systems theory, a book written by this man, Ludwig von Bertalanffy. And he was inspired by his study of biology, and he was trying to come up with a term to encompass the understanding of organismic science he had seen as a biologist, because biology is very complicated, and I'll talk about that a little bit more. So for the purposes of this talk, I'm going to separate biology and systems biology into a few different dimensions, and those are the molecular, structural, and temporal complexity. So the molecular complexity actually refers to all of the small molecules and the proteins, the DNA, and the genes, which make up the organism as a whole. The structural complexity is how all these little bits and pieces stack together to make the three-dimensional structure that you see when you look at a cell, both with the organelles and the protein structures inside and the outer membrane also. So, and the temporal complexity really kind of engulfs both the molecular and the structural complexity and the dynamic changes that happen in these over time. So we obviously can't take and look at every part of a cell and model it and understand what's going on. So often what we do is we take and we abstract things, and we come up with abstract models where we take small pieces of the cell, and we try to make simplified models to explain what we see. And so here's a little schematic that I designed for Yang in McDermott's lab, where he showed that the metabolisms of arsenic and phosphorus are put together, phosphate and arsenic metabolisms are put together. You can see that these regulatory networks are interrelated, but we don't really come up with a holistic view of the cell, and we don't really have a mathematical model. We know that certain things cause other things to react in certain ways, like an increase in abundance of levels and a decrease in abundance of levels, but we're really just trying to take one small piece and digest that at a time. So how do we do and generate these models? We use something a lot of times called omics technology, and it allows us to view both the molecular and the temporal complexity if we design these experiments correctly. And omics can actually refer to a variety of different subdisciplines. Genomics, where you look at the genes in the DNA, the transcriptomics, where you look at the RNA and its abundance, the proteomics and the proteins, all of the proteins and its abundance, and the metabolomics and the metabolites and all of their abundance. So, to give you a better grasp of the complexity, if you look at the DNA that exists in a human being, you actually have about the latest hypothesized number are 25,600 genes, and then even a simple bug like E. coli has around 5,000 genes. In humans, it's proposed that there's 20,687 protein coding genes that make up individual proteins. And each one of these proteins can undergo post-translational modifications or a variety of different modifications. So this number is probably orders of magnitude higher for the number of different proteins in the cell. E. coli, you can see, has a little bit lower number because it's a very simple bug. And then if you look at the metabolites that exist in the human, in the HMDB database, there's actually 41,514 metabolites that have been seen. So when you take all of this data in, you just kind of understand just how complex these organisms are, or we are, at a molecular level. And our lab, in specifically, we kind of focus on Sulfolobus sulfotericus, and we've used it as a systems model. And Sulfolobus actually is a hyperthermophilic archaea. And so if you look here, we have a phylogenetic sequence where we split the domains of life into the archaea, the bacteria, and the eukaryotes. So you're over here. And so we expect organisms to be different based upon where they lie on this tree. So organisms that are more closely related actually sit on closer branch points, and organisms that are more distantly related, there's more distance between them. And you can see I've actually colored this to show that the organisms at the base of this tree are actually thermophilic organisms, where the red is the higher temperatures, the yellow is more moderate, and the blue is a little more cool. So the first thing that I actually worked on in the lab as an undergraduate was Sulfolobus as a systems model for virus infection. So we looked at Sulfolobus and how it responded to virus infection with this virus here, Sulfolobus turritid icosahedrovirus. And in the process, came up with and discovered a protein that wasn't related to the virus that was infecting it, but another virus-like protein that seemed to be found in the genome of Sulfolobus, and the protein fold was similar to a bunch of viruses that infect the bacterial domain of life. So a little bit of background, there's actually a set number of viral lineages, so like the eukaryotic herpes virus and the HK97 viruses that infect bacteria are actually thought to be closely related through both their structural and their functional similarities. And so when you have virus lineages and viruses that are closely related, you can look at how they're related by how closely their protein sequences or their DNA sequences are to one another. But you can also, when the relationship is a bit more divergent, you can look at more highly conserved features like structure and function. But that requires a little bit more like crystal structure solving, so you can actually compare the structure of these protein sequences. So viruses have influenced the evolution of probably all life through the transfer of genetic elements and the synthesis of novel host proteins. So I'll talk about that a little bit more, but the thing to take away from this is viruses can actually switch tropisms. So you'll hear in the news where you'll have swine flu that switches over to humans and it becomes more toxic. But actually what can happen is these viruses can switch tropisms and go from one organism to another, and they can actually transfer DNA between these different organisms. And then through their recombination events, they can actually make novel proteins that have different functions, and they can introduce these new genes into the new organisms, and it's thought to be a pretty prevalent driver of evolution itself. So a review of kind of what happened in the study is during the STIV infection, we spotted this protein right here, which was actually just annotated as SSO 2749, and it was upregulated during virus infection. And it turned out that this protein was very similar to a virus protein that had been discovered in a similar organism. And so I modeled this protein and just made a nice little visual here. And it was proposed to stack into these virus-like particles. And so when we actually took a look inside of Cephalobus, we could see these virus-like particles. And this is a TEM image, thanks to Sue Brumsfield, who helped me put it together. So this protein was actually pretty interesting from a couple different aspects. And the first was, not only was it similar to this other protein in this similar organism, Pyrococcus furiosus, but it also was had a sequence level similarity to these encapsulin proteins that were found in Thermatoga. And these encapsulin proteins weren't thought to be part of a virus at all, but they did have structural similarity to the virus. And it was structurally similar to the HK97 virus GP5 major protein that infects bacteria that I talked about in the beginning. So when I looked at, so the next step for analyzing this protein was kind of to look at the genomic context. So I wanted to go into the genome and I wanted to look and say, okay, where does this sit, and can I figure out anything about what this protein might do based upon what it's close to? Because often you'll have proteins that will be transcribed, or RNA which will be transcribed in sequence. And so when I looked, I could actually find a variety of different proteins that had similarity to a bunch of different viruses from different domains of life near this protein. And it turned out I could find an attachment left and right site for this virus-like particle. And I had a bunch of unknown host proteins kind of stuck in between them. And so this is interesting because when certain viruses in their life cycle, they have a lytic and a lysogenic phase. So viruses will go and they will infect a host, and then they'll actually stick their DNA into the host genome, and it'll just sort of sit there dormant for a long period of time. This is called the lysogenic cycle, until something comes and stresses, usually starts killing the host, and it triggers the virus particles to start being made again. And so this helps you sort of understand how these viruses can add to the genome of the organisms that they infect. And this is pretty interesting because it has pretty big implications for how virus proteins can be used by the host genome, and how that they potentially could be adopted by the host for certain functions. So I talked about the encapsulin protein. I'm sorry about that. The encapsulin protein, and it turns out that it could be possible that the encapsulin protein actually originated from a virus, and while most of the virus studied field, and it's a little bit newer than the other three. So, omics, what is it good for? So, can you generate a mathematical model using just omics data? No, you can't, because you're just looking at abundance levels. You're not looking at flux. All you see is patterns. You're seeing patterns of things that go up, and you're seeing patterns of things that go down. And it turns out that there's some really cool machine learning pattern recognition software that's been used in a myriad of different things in life, and some of it is facial recognition. So the FBI likes to look at different pictures of people's faces, and they have a pattern recognition algorithm that tries to spot when they can see, a convicted felon or a spy or whatever and identify these. But it turns out you can actually use these machine learning pattern approaches to find patterns in metabolism also. And so this can be particularly useful because metabolic patterns can reveal biomarkers of disease and stress. So when you go to the doctor and the doctor draws your blood, he's looking at certain things in your blood, and those things could be referred to as biomarkers. So he's saying if your cholesterol is too high, this could be an indication that you're going to get heart disease soon or later on in life. And so what we can do is we can take a limited number of biological samples, and we can try and spot the small molecules that are indicative of like a disease or a stress. And we like to find ones that are very robust, so we can just study a few animals. We don't have to study a whole lot. And we can apply those biomarkers to larger populations. So this actually led to this publication, which I call the Application of Support Vector Machines to Metabolomics Experiments with Limited Replicates, which will make a little more sense in a few minutes. So this was actually in collaboration with Aurelian, who did all of the data analysis, and then Monica, who ran the untargeted metabolomic analysis on the pigs. And we kind of looked at our two model systems, and we looked at them after a certain stressor. So in Sulfolobus's case, we looked at it before and after oxidative stress, and in the pig's case, we looked at it before and after hemorrhagic shock. So what's the good thing about metabolomics? Metabolite profiles, and so that's all of the abundance levels of the metabolites themselves, are sensitive to a wide variety of environmental influences. And you might be able to say that they are sensitive to almost every environmental influence. And so they're really good for finding biomarkers. And so the bad thing about metabolomics is you often have limited sample sizes. So you can't go and take 100 people out of the population and try to find biomarkers that are going to apply to 6 billion people, right? And so you're trying to model what other people's bodies are doing by just looking at a few. And so there's actually a few problems with these sort of data sets, and that the first is there's very few biological samples. So you're usually looking at less than 10 organisms, and you have a whole lot of metabolites that you're looking at, and so on the order of thousands. And so this means that machine learning really isn't optimized for these sort of data sets. But there's a special little algorithm called support vector machine recursive feature elimination that tries to make machine learning a little bit more applicable to these sorts of data sets. And what it does is it eliminates the noisy features and tries to look at maybe 10 different biomarkers at once, or 10 different metabolites at once, instead of thousands, by ranking them. And I'll talk a little bit more about those in a few minutes. So, as I said, we use two model systems for evaluating the metabolic response to stress. And the first one was Sulfolobus and its stress to oxidative stress with hydrogen peroxide, and the pig and its stress to shock. And then we kind of look and see what goes up and down in all of these. So you don't have to understand how machine learning works. Like, this isn't a PhD defense for machine learning or computer science. All it does is it sort of can recognize different categories that an organism can sit in. And so you break your data sets into training data sets and testing data sets. So let's say you have 10 organisms. You'll take 5 of those organisms and you'll train the support vector machine. And then you'll take 5 and you'll test. And then it's evaluated based on its accuracy. If it can accurately classify 5 out of 5, it's 100% effective at discriminating. If it's 4 out of 5, it's 80%. Now, we use this in conjunction with the recursive feature elimination, and so the SVM is actually run many times, and it ranks all of the metabolites and removes those that are less informative or noisy. And so, like here, if you look at this metabolite profile, it would go in and it would say, you're the best at discriminating between the oxidative stress and the healthy, or the hemorrhagic shock and the healthy, and you're the second best, you're the third, you're the fourth, you're the fifth, and it knocks out the rest and says, I don't even want to look at you because you just confuse me. So let's look at the first data set. So when we first analyzed Sulfolobus, we had 10 baseline replicates and we had 10 stressed replicates. And if you look at this data, and so all you have to take from this figure is this is the coefficient of variation, and you can see that the histogram shows that most of the features are here at the left side of the axis, which means that they're very similar. There's not much difference in these data sets. All of these bugs are really closely dealing with this oxidative stress. And if we use a very simple pattern recognition algorithm, so this clustering, we can see that it's really easy for an algorithm to say whether it's a healthy baseline or a stress, so it can really group those. So it almost is not reasonable to use the support vector machine on these, except for when we compare the two data sets. So what happens is we take our full range, our 10 and 10 replicates, and we train the support vector machine, and then we evaluate its accuracy. So it's actually right here. This is the axis. I blew it up so you could see it. So it's 100% accurate. Then we go down to 8, and then we test with 4, and we're still 100%. And you can see that even when we reduce the number of replicates, because these organisms are so similar, we never really drop below 100% accuracy. The support vector machine can tell every time whether these organisms are healthy or whether they are diseased, or stressed, excuse me. But let's look at the pigs. So we actually evaluated pig urine, which is known to be pretty noisy. And so you look at this same diagram again, and you can see that the axis is actually shifted, and the histogram is more here in the middle. And it's showing that these pigs are actually dealing with this stress and are actually quite different from one another in comparison from Sulfolobus. When I try to do a simple pattern recognition algorithm, you can see that I have black mixed with these red dots and some more black up here. So it kind of confuses the more simple algorithm. And then when I reduce the number of replicates, I'll have you just look at this gray line first. You can see that when I go from the full scope and I start reducing the number that I train the support vector machine, we actually drop from 100% at the 6 and 6, and we go down to below 70%, which actually is a pretty poorly trained support vector machine. But what happens is when we run our recursive feature elimination and we just take these top-ranked features and we eliminate the noisy ones, we have this new blue line that shows that, bam, it's 100% accurate even when we're only looking at 4 different pigs. And it can tell with the 16 other pigs, or excuse me, the 12 other pigs, it can say 100% of the time whether they have been diseased or stressed. And the other thing to look at is here we have the metabolite rankings. And so the green are the top 1%, the purple are the next 5%, 10%, and then so on. And so these green ranked biomarkers are actually pretty well conserved, and they retain their predictive ability even when the small data sets, which suggests that they are probably pretty good biomarkers if you wanted to look at an entire population of pigs instead of just 10. The SVM was accurate at predicting what the group metabolite data sets belonged in, and the SVM RFE increased the accuracy when the number of the training metabolite data sets was low, which, right, is important because we're obviously not testing the entire population. And the SVM RFE could find accurate biomarkers using limited metabolite data sets, and is probably good for prediction of larger populations.
Can you say briefly what the reviewers have been complaining about? You say that the reviewers are too happy with this approach. I mean, I don't want you to go into detail, but just a couple of comments, because it looks pretty good.
The first round of reviews, it seemed like it was pretty trivial, and then we got comments from the third reviewer, which had never come in. So we only got the review from the first two reviewers, and then the third one came in, and they're like, well, you didn't do a good enough cross-validation. So what you do is you do it either a leave one out cross-validation, and that's what we did. And he said, a leave one out might not be good. You should probably do an n-fold cross-validation. And so we had to go through multiple rounds of review. So the first comments I felt were pretty minor, and then this last round was just kind of a pain because doing these recursive calculations takes weeks and weeks. So we're really going to run them on the cluster, and I think it took like 2 weeks and probably millions of runs to do this. And in the end, it ended up being almost exactly the same results. So it was kind of a waste of time.
But it makes it stronger, I guess.
Sure. And so one of the future directions that I would like to go in, and I haven't really been able to yet at this point, was a multi-class classification. So I looked at sulfolobus under a myriad of different conditions, and I tried to categorize it all in all of these different categories. And when I looked at data sets where I had 6 different conditions and 60 different data sets, it was 98.33% accurate, and that was without recursive feature elimination. That was just an accuracy analysis. So for my last little section here, I call it the Future of Omics Stage 2, and that's studying metabolism over time. So after running a bunch of metabolomics data sets, you realize how long and laborious it can be, and it takes a long time to generate good data. And because you have to, culture cells or extract your biofluid, like your blood or your urine, then you have to extract the metabolites from there and separate it from the proteins and purify it. And then you have to run, each replicate is at least 30 minutes of chromatography. And so, realistically, when you do one of these experiments, it takes weeks. So you're looking at a limited set of samples, and you're spending weeks trying to get the information out of these. And, the changes that occurred in metabolism over time first became apparent to me when I was looking at sulfolobus, and I did this little time course between 15, 30 minutes, 60 and 105. And then I tried to group them, and you can see it's just kind of little mud here. So if this was working how I would ideally like to see it, you would see little groupings, like the orange would all group together, the red would all group together. But it turns out they turn into like this muddy mix over time. And it's even worse when you look at longer time periods, like 105 minutes, 8 hours, and 16 hours. You can see that none of this stuff groups well at all. So it came to my last little study, which I titled Real-Time Digitization of Metabolomic Patterns from a Living System. And this was actually done with a lot of help from Mohammed in the microfabrication facility. And they made a little microfluidics chip so I could extract metabolites in real time. And I'll show you.